Am I audible to all?
Yes, sir.
Please write your enrollment number in the chat box. Please write your enrollment number in the chat box. All you write your enrollment number in the chat box right now. Only 6 students have written. Attendance can be recorded. Have you all written? Have you all written your number?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. 1, 2, 3, 4, 5. Please write your enrollment number in the chat box. Only you have written, only 7 to 8 students have written. And those who have not written your enrollment number, please write in the chat box. Because attendance is so much essential for us. Get that? And 10 students have already joined. And now we'll start our topic. And those who have not written, please write it. Hello. Are you getting me?
Yes, sir. Yes, sir.
And then who have not written?
Yes, sir.
Who have not written the enrollment number? Okay now. And now okay. Hello. And now
Yes,
and
sir.
we'll start our the topic. Okay.
Yes, sir.
Yes, sir.
Okay, now, and we have start without the now the new chapter, that is of the respiration. So, then what's your idea about the respiration? What's your idea about the respiration? What's all your idea about the respiration? Anyone? And if anyone asks you the respiration, actually, what do you understand?
Excuse me, sir. Little more volume. I'm not able to that much audible.
I'm not audible to you all. Hello.
Little
Now I'm
more
audible.
volume could be there. Nice.
I'm audible right now.
Yes, sir.
Okay. So let's, and we have to discuss with the respiration. And then the word respiration has been derived from the Greek word, sorry, and from the Latin word, that is the respirare. Respire means to breathe, means inspiration and expressions is comprising of the respirations. Got this point? Hello.
Yes, yes, sir.
Okay. So that here, and then and overall when you speak, the process by which organism take gas and release the gas is known as the respiration. And the types
Sir,
of the
voice
respiration.
ଶୁଭୁନି। Sir, sir ଶୁଣା ଯାଉନି।
Subhune, just a minute.
ନା।
Sir, voice clear ଆସୁନି।
Okay. And just a minute. Now I'm audible to you.
Yes, sir.
Hello.
Yes, sir.
Sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay. Your students are coming continuously. Who have come lately, please write your alarm number in your chat box. Thereby your attendance can be recorded. Do write. Okay. So first we have to start with the types of the respiration, and only understand the exchange of the gas is technically known as the respirations. And we are having three types of the respiration. One is the external respiration, internal respiration, as well as the cellular respirations. And always try to understand this topic very carefully. So now first we discuss about what is external respirations. So that external respiration is a purely physical process which involves exchange of the gases, especially oxygen and a carbon dioxide at the organismal level. For example, and then when the organism exchange the gas with the environment is known as the external respiration. For example, this is the nose, the process we are taking oxygen and releasing carbon dioxide. This is known as the external respirations. For this point, naka bate oxygen nelle chadile carbon dioxide, it is the external respirations. So external respiration operates at the organismal level. Got this point?
Yes, sir.
Yes, sir.
Next come to the internal respirations. And in case of the internal respiration, what happen? It is the exchange of the oxygen and carbon dioxide at cellular level, or when the cell exchange oxygen and carbon dioxide with the environment is known as internal respiration. For example, and first we are having the external respiration. By the process of external respiration, oxygen now through the nose goes into the lungs. Then from the lungs through the blood, the oxygen is carried to the cell. Now the oxygen enter into the cell. Now inside the cell, when oxygen entered, is converted into carbon dioxide. Again, carbon dioxide enter into the blood. So the process of exchange of oxygen and carbon dioxide at the cellular level is known as the internal respirations. Then from the external respiration and from the internal respiration, we have no idea how this oxygen is converted into carbon dioxide. Understood this topic? This point?
Sir, cellular respiration टा आपण कहि ना ना.
No.
Okay, sir.
Next come to our cellular respirations. So cellular respiration can be defined as, look at, in case of the external respiration as well as in case of the internal respiration, then this oxygen is converted into carbon dioxide, but we have no idea how this oxygen is converted into carbon dioxide. So to understand how this oxygen is converted into carbon dioxide, so first we have to take the help of the cellular respirations. Look at, next come to the cellular respirations. Cellular respiration is the biological process of oxidation of food material by using molecular oxygen, producing carbon dioxide and water, and releasing energy in small steps and storing it in a biological useful form, that is the ATP. For example, so by the process of external respiration, organism take oxygen, then this oxygen is now go to lungs, then to the blood, then finally enter into the cell. Then inside the cell, this molecular oxygen now react with the glucose. Glucose is the food material. Now this molecular oxygen is now react with the glucose. So in presence of the oxygen, this glucose molecule become breakdown to form 6 carbon dioxide, 6 water, and energy. And nearly 60% of energy become lost in the form of heat, and the rest of energy stored in the form of ATP. And the carbon dioxide that we are releasing, and through the nose, this is the carbon dioxide that is released due to the breakdown of glucose by the oxygen. By this point, so this is our cellular respiration. Cellular respiration means the oxygen we are taking, this oxygen finally come into the cell. Inside the cell, this oxygen react with the food material. So food material break into carbon dioxide and water. This carbon dioxide finally enter into the blood, then to the lungs, finally to the nose, it comes out. Got this point?
Yes,
Yes, sir.
sir.
Now here,
Sir,
now, okay, speak.
sir, आमे जो aerobic respiration और anaerobic पढ़िथिलु तार आ सहित किछ रिलेट नै सर.
ना ये तम तो एटा तो एरोबिक ना, एटा ऑक्सीजन यूज होची हैला से गोटे तम होला कोन, दिस इज द देन दिस इज द टाइप ऑफ द रेस्पिरेशन ऑन द बेसिस ऑफ प्रेजेंस एंड एब्सेंस ऑफ ऑक्सीजन. So you are the aerobic, anaerobic, and fermentation. We are having 3 types of respiration: aerobic, anaerobic, and a fermentation. When respiration occurs in presence of molecular oxygen, is known as aerobic. When respiration occurs in presence of oxygen but not in free form, is known as anaerobic. When respiration occurs in complete absence of oxygen, is known as the fermentation.
Yes, okay.
So look at, now we are dealing with our cellular respiration. We have to discuss, and then look at, and by the process of cellular, and then here to explain cellular respiration, we have to take the carbohydrate respiration. Here the food material is the carbohydrate. So now the glucose, when the glucose react with the oxygen, here it should be 6 oxygen. Here this glucose react with the 6 molecule of oxygen to form 6 carbon dioxide, 6 molecule of water, and a 38 molecule of ATP. So not in a single step, a molecule of glucose react with 6 molecule of oxygen to form 6 carbon dioxide, 6 water, and a 38 molecule of ATP. And it could not be occur in a single step. So this conversion require the 4 steps. So our cellular respiration is completed through 4 steps. One is glycolysis, 2 is the oxidative decarboxylation of pyruvic acid, Krebs cycle. The fourth one is electron transport systems. Then after a glucose molecule pass through this 4 step, finally a glucose molecule completely oxidized, that causes release of 6 molecule of carbon dioxide, 6 molecule of water, and a 38 molecule of ATP. So first we have to understand, and before we enter into the mechanism of all these steps, so first we understand the overall mechanism. Actually, what happened in the glycolysis, what happened in the oxidative decarboxylation of pyruvic acid, actually what occurs in the Krebs cycle, and what occurs in the electron transport system. So look at, now this is the glucose. The glucose is present inside the cell. So, and then the first step of the respiration is the glycolysis. And then, and now first, and if when the glucose enter the respiration, it has to be encountered the glycolysis, or the first step is the glycolysis. When a glucose molecule enter the glycolysis, what happen here? This glucose molecule contains 6 carbon atom. This glucose molecule becomes split between carbon atom 3 and carbon atom 4. Glucose molecule dikhonda hegala and converted into 2 molecule of 3 carbon compound known as pyruvic acid. This pyruvic acid is a 3 carbon containing compound. When this glucose molecule is break into 2 molecules of 3 carbon compound, at the same time the 4 molecule of ATP and 2 molecule of NADH plus become released. At the same time 2 molecule of ATP also consumed. Here I have not written there. Okay, look at, and finally the 2 molecule of NADH plus S plus released, 4 molecule of ATP released, and the 2 molecule of ATP has been consumed. So by the process of the glycolysis, the net gain of ATP is 2. Got this point? 4 ATP bahala, 2 molecule ATP use hoichi, tale kete net gain 2. Hello.
Yes, sir.
Yes, sir.
Look at, and then, and when the glucose undergo oxidation, mane kon na glycolysis glucose bhanguchi, the breakdown of the complex molecule into simpler molecule known as oxidation. When the glucose undergo oxidation, that causes the loss of electron. This electron is accepted by the NAD plus, that will become converted into NADH plus S plus. Always you have to understand, when the glucose breakdown, when the glucose breakdown, the electron become released, that will become accepted by the NAD plus. So NADH plus, now the NAD plus become NADH plus S plus. So NADH plus S plus is always the source of 2 electrons. Next come to the pyruvic acid. Now here, this particular 2 molecule of pyruvic acid that become present here, they undergo oxidative decarboxylation. Oxidative decarboxylation means you have to understand oxidation. Oxidation and decarboxylation. Oxidation means the loss of electron or loss of hydrogen atom. Here the decarboxylation means loss of the carbon dioxide. Here the pyruvic acid contain our 3 carbon atom, or the pyruvic acid contain 3 carbon, and here by the process of decarboxylation, this pyruvic acid loss 1 carbon in the form of carbon dioxide, and also it undergo oxidation means lose the 2 electron. After losing the 2 electron and 1 molecule of carbon dioxide, the phosphoric acid become acetyl coenzyme A. The pyruvic acid become acetyl coenzyme A in conjunction with the coenzyme A. So बुझेला ए लाके ना pyruvic acid कोन जो एबे ना decarboxylation गला, गोटे कार्बन डाइऑक्साइड लॉस कला, and under oxidation गला दीटा इलेक्ट्रॉन लॉस कला. So electron become accepted by NAD plus, so NAD plus become NADH plus S plus, and a carbon dioxide release. After that, the pyruvic acid has lost the one carbon in the form of CO2, and now the remaining part left is the acetyl group. This acetyl group react to the coenzyme A to form acetyl coenzyme A. Here the number 2 is there because there is 2 molecule of pyruvic acid, So 2 molecule of acetyl coenzyme may form. Got this point?
Yes, sir.
Look at, and then the 2 molecule of acetyl coenzyme that present here at the end of the second step. Then 1 molecule of acetyl coenzyme is required to carry out 1 Krebs cycle. We are having 2 molecules of acetyl coenzyme, so there should be 2 cycle. Now, when 1 acetyl coenzyme enter one Krebs cycle, that cause release of 3 molecule of NADH plus H plus, 1 molecule of FADH2, and 1 molecule of ATP. Now, and we are having 2 molecule of acetyl coenzyme. For that, we are having 2 Krebs cycle. So 6 molecule of NADH plus H plus, 2 molecule of FADH2, 2 molecule of ATP form. For this point, now after the Krebs cycle, the carbon of the glucose is completely oxidized. Means here, look at, and in this step also, the 4 carbon dioxide become released. Here I have not mentioned there, and to make the picture very simple. So here, in case of the third step, that is our Krebs cycle, here also 4 carbon dioxide become released. In case of the respiration, the 2 carbon dioxide released in the second step, and the 4 carbon dioxide released in the third step. So there is nothing is left at the end of the TCA cycle or Krebs cycle. बुझिला point एबे? Hello.
Yes, sir.
Look at, and here, look at now we are having 3 steps have been completed. How many molecule of ATP formed it? 2 molecule of, and here the 3 steps have been completed. How many molecule of ATP formed it?
6.
Look at there. How many? 4 and 2. 6 molecule of ATP formed. How many molecule of ATP need to be formed?
38.
How many molecule of ATP now need to be formed?
2.
32.
32.
Now you have to look into actually where these 32 molecule of ATP present. एबे को जगह से 32 molecule of ATP अछंती. Look at. Then 1 molecule of NADH plus is equivalent to 3 molecule of ATP. 1 molecule of FADH2 is equivalent to 2 molecule of ATP. And this is applicable for the NADH plus that formed in the second step, that is the oxidative decarboxylation of pyruvic acid, and is applicable to NADH plus that formed in the Krebs cycle. But the 2 molecule of NADH plus that formed during glycolysis, then each molecule can produce the 2 molecule of ATP, or the NADH that formed in the glycolysis is equivalent to 2 molecule of ATP. Now count and tell me, and look at, and when this NADH, H plus and FADH2, look at, all are the source of electron. When this NADH, H plus and FADH2 and they transport their electron to the electron transport system, in this case they form ATP. So 1 molecule of NADH is equivalent to 3 molecule of ATP, and a 2 molecule of FADH is equivalent to 2 molecule of ATP. 1 molecule of FADH is G and 2 molecule of ATP. Now with respect to this calculation, tell me how many molecule of ATP will be formed in ETS? How many molecule of ATP will form in ETS?
सर बुझा पड़लानी किछे.
देखो एटा. And then now here the 3 steps have been completed. 6 molecule of ATP formed. Actually where are remaining 32 molecule of ATP. Here the 32 molecule of ATP are present inside NADH, H plus, H plus and FADH2. When NADH, H plus, H plus and FADH2 and undergo electron transport system, because they are source of electron. When they transfer the electron into the ETS or electron transfer system, in this case they can form ATP. Then if 1 molecule of FADH2 is equivalent to 2 molecule of ATP. गोटे FADH2 भितर भाबी देबो 2 ATP अछि. गोटे NADH भितर भाबी देबो 3 ATP अछि. तार तो ई जो हमें जो कहूचे जे जो ग्लाइकोलाइसिस NADH अछि, प्रत्येक NADH हमरो ना हमरो only 2 molecular of ATP अछि और this the NADH plus of the glycolysis is equivalent to 2 molecular of ATP. So count then how many molecule of ATP will release by the process of PTS. Ready? FADH into 2 4
हेले से
एटे कोन
18 4
6टा NADH
22
अछि 18टा हबो ऊपर केते अछि NADH प्लस अछि 3टा 6टा ग्लाइकोलाइसिस जो NADH प्लस अछि तार कोन हबो तार भारिबो 4टा हे गेला केते 32 हे गेला 32 होला परे या गोबर 6टा तुम देख केते एबे 38 हे जिवो तो बुझिला
सर NADH रे 3टा ATP पर सर
for the second step and third step but glycolysis mean 2 ATP.
ओके
Okay माने कोन पई then we have discussed and when study about ETS. Got this point? So
Yes,
now
sir.
we have to discuss about the glycolysis right now. Sorry, now come to about the site of respiration. Actually, where these 4 steps occur inside the cell. Look at the glucose present here in the cytoplasm. So the glycolysis occur in the cytoplasm. After the glycolysis occur, what result? What is the end product of the glycolysis?
Pyruvic acid.
Pyruvate. Now this pyruvic acid enter into the mitochondria because second step, third step, and fourth step will occur inside the mitochondria. Now this pyruvic acid enter into the perimitochondrial space or intermembrane space of the mitochondria. This is outer membrane, this is the inner membrane. This space is known as perimitochondrial space. Now this become enter here. Now look at the outer membrane of the mitochondria is very freely permeable to any molecules and any molecules can easily cross. But inner membrane of mitochondria is highly rigid, is highly impermeable. That अर्थ ये inner membrane भितर को कहीं पस परिवे नहीं. Even a single proton cannot enter into the matrix through the inner mitochondrial membrane. But pyruvic acid is a large molecule, so pyruvic acid cannot enter. Always you have to consider there is high concentration of H plus present in the perimitochondrial space or in the intermembrane space. Here, how this pyruvic acid become enter? When this pyruvic acid become enter, the pyruvic acid, now look at, now we are having a pyruvate symphoter which present in the inner mitochondrial membrane. This pyruvate symphoter take one molecule of pyruvic acid and one H plus and transport into the matrix. After the pyruvate transport into the matrix, the pyruvate undergo second step. In case of second step, what happen? The pyruvic acid undergo oxidative decarboxylation and a combine with the coenzyme to form acetyl coenzyme. After that, acetyl coenzyme enter the Krebs cycle, and after the Krebs cycle, nothing is left. Only FADH2 and NADH H plus are left. This NADH and H plus and FADH2 they undergo electron transport system. The component of the electron transport system present along the inner membrane of the mitochondria. Look at when this NADH H plus and FADH2 they transfer the electron through the electron transport system and then that causes formation of the ATP. For this point,
Yes, sir.
so now glycolysis occur where?
Outside the mitochondria.
Glycolysis occur in the cytoplasm or in the cytosol. Then second step inside matrix, third step inside the matrix or the Krebs cycle occur in the matrix, and the electron transport system takes place where? Electron transport
Inside
system
the
takes place where? Not inside.
in
Along the inner membrane of mitochondria. Look at. Okay. Because the electron transport system is consist of 4 complexes: complex 1, complex 2, complex 3, and complex 4. So the glycolysis occur in the cytoplasm or in the cytosol. Second step, third step occur in the matrix, and the fourth step occur along the inner membrane of the mitochondria. Clear this point?
sir.
And who have joined lately, please write your enrollment number in the chat box. Okay, now come to the glycolysis. So that the glycolysis is a sequence of biochemical reaction in which a glucose molecule split into two molecules of pyruvic acid, as you know, with the release of our 4 molecule of ATP and 2 molecule of NADH plus H plus. The process of glycolysis was discovered by Gigi Embeden, Otto Meyerhoff, and Jacob Parnas. So this pathway also known as Embeden-Meyerhoff-Parnas pathway or EMP pathway. Now come the steps of the glycolysis. And always you have to remember that glucose is a very highly stable molecule. So the glucose will not participate directly in the glycolysis. And if we say glycolysis is a chemical reaction or is a reaction, glucose is a reactant. So glucose do not directly participate in the glycolysis because glucose is a very highly stable molecule. Before this glucose participate in the glycolysis, this glucose need to be activate. Then how a molecule become activate? Any idea? How a molecule become activate? Any idea of anyone? How a molecule become activate? In general sense, by getting energy. Okay, a molecule become activated by getting energy. So before this glucose molecule enter the glycolysis, this glucose molecule need to be activated by supplying energy. So ATP is the source of energy inside the cell. Look at the process of glycolysis is completed by our 10 steps. The 10 steps are divided into our the look at into the two phases. One is preparatory phase, second is the payoff phase. So first step we will discuss about the preparatory phase, and the first step is the phosphorylation. Here the preparatory phase which makes the glucose ready to release ATP in the payoff phase. Always you have to remember. Look at in the first step what happened. This glucose is a highly stable molecule. Now this is the structure of the glucose having 6 carbon atom. Look at in first step what happened. The glucose react to the ATP in presence of enzyme hexokinase to form glucose 6-phosphate. Glucose 6-phosphate means glucose has not 6-phosphate, but the 6 carbon of the glucose is attached with a phosphate. ATP having 3 phosphate, and after transfer 1 phosphate to the glucose carbon number 6, ATP having 2 phosphate, so ATP become released and we are having glucose 6-phosphate. Got this point? Then this is about activation glucose. Hello?
Yes, sir.
Look at in case of the next step what happened, this glucose 6-phosphate undergo isomerization in presence of enzyme phosphohexose isomerase to form fructose 6-phosphate. In the next step, again phosphorylation means addition of phosphate group from ATP here. So here this fructose 6-phosphate now react with the ATP in presence of enzyme phosphofructokinase to form fructose 1,6-bisphosphate. It means then look at the carbon number 6 of the fructose, there is a phosphate group present, and the carbon number 1 there is a phosphate group present. So fructose having the 2 phosphate at carbon number 1 and carbon number 6. So this fructose 1,6-bisphosphate. Now come to the step 4, that's cleavage. Now this fructose 1,6-bisphosphate break between carbon atom 3 and 4, this position, so to form 2 molecule of 3 carbon compound. No, then one is known as glyceraldehyde 3-phosphate and second is known as the dihydroxyacetone phosphate. बुझिपाइलो एबे कोन फ्रुक्टोज 1,6-bisphosphate भांगी जीवो, भांगीले कोन हबो ग्लिसरल्डिहाइड 3-phosphate हबो आउट ऑफ डाइहाइड्रोक्सीएसीटोन फॉस्फेट हबो. बतमे ए जो डाइहाइड्रोक्सीएसीटोन फॉस्फेट हबो से कोन करबो ता पर स्टेप रे चेंज इनटू ग्लिसरल्डिहाइड 3-phosphate because the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are isomer of each other, and here dihydroxyacetone phosphate now change into glyceraldehyde 3-phosphate. Now we are having how many molecule of glyceraldehyde 3-phosphate right now? We are having how many molecule of glyceraldehyde 3-phosphate right now? वर्तमान हम पाखे केतेरा molecule of glyceraldehyde 3-phosphate अछि पिला? हेलो
sir.
केतेटा माने वर्तमान 5 नंबर स्टेप परे हम माने पके केते मॉलिक्यूल ऑफ ग्लिसरॉल्डिहाइड 3 फॉस्फेट अछि
3टा
3टा अछि अरे मूर्ख ऊपर तो गोटा बाहर छी गोटा बाहर तला ऊपर 5 4 नंबर रे आ गोटे वाला 5 नंबर रे ऊपर वाला कोन गोटे ना ग्लिसरॉल्ड 3 फॉस्फेट गोटे बाहर तला डाइहाइड्रोक्सी फॉस्फेट जोटा बाहर तला डाइहाइड्रोक्सी फॉस्फेट सेटा चेंज हे गेला पुनी ग्लिसरॉल्डिहाइड 3 फॉस्फेट केतेटा अछि
दीटा
ला देखो एबे असलम पे ऑफ फेज एबे कोन कले ग्लूकोज को हम एक्टिवेट के रखिले एमसी पर एमती हमर एनर्जी बाहर करियो एबे पे ऑफ फेज माने जोटा को प्रिपरेटरी फेज पे ऑफ फेज एडा एला अमल फेज हमर आर फर्स्ट एबे कोन डिहाइड्रोजिनेशन एंड फॉस्फोरिलेशन हियर डिहाइड्रोजिनेशन मींस ऑक्सीडेशन एंड फॉस्फोरिलेशन इन दिस स्टेप व्हाट हैपन this particular glyceraldehyde 3 phosphate undergo dehydrogenation means it lose 2 electron and 2 proton that become accepted by NAD plus so NAD plus become NADH plus H plus as well as and here it react with the phosphate molecule so look at as you know that when aldehyde become oxidized to form the acid so due to of the oxidation of the aldehyde so here now we are having glyceraldehyde there. Now this when this glyceraldehyde become oxidized to form glyceric acid, but here we are having glyceraldehyde 3 phosphate. When glyceraldehyde 3 phosphate become oxidized to form 3 phosphoglyceric acid, as well as the 2 molecule of NADH plus H plus become released here. So this the 3 phosphoglyceric acid now react to the phosphate now react to the phosphate group. Now this phosphate group attached with the carbon number 1. So to form 1,3-bis phosphoglyceric acid or glycerate. Got this point? Hello.
Yes, sir.
ये देखो फर्स्ट कौन है this glyceraldehyde 3 phosphate first undergo oxidation to form 3 phosphoglyceric acid or glycerate plus or NADH plus H plus. Then the extra phosphate now here react with this particular carboxylic group to form 1,3-bis phosphoglyceric acid or glycerate plus look at here the 2 implies we have 2 molecule of glyceraldehyde means the 2 then the 2 molecule of 1,3-bis phosphoglyceric acid. Look at now at the step 6 we are having 2 molecule of 1,3-bis phosphoglyceric acid or glycerate. In the step 7 is the active generation and in this step what happened our the 1,3-bis phosphoglyceric acid having 1 phosphate at carbon 1 1 phosphate at carbon 3. Look I'm having 1 phosphate at carbon 1 and 1 phosphate at carbon 3. So it transfer 1 phosphate to the ADP having 2 phosphate when ADP get 1 phosphate it become ATP. Now it again become 3 phosphoglycerate or 3 phosphoglyceric acid. After that, the 3 phosphoglyceric acid undergo isomerization to form 2 phosphoglyceric acid. It means phosphate from carbon number 2 now shifted to carbon number from the carbon number 3 becomes shifted into carbon number 2. Okay, we want 2 phosphoglycerate or glyceric acid. The next step is the dehydration. Dehydration means what? The release of the water. Now the 2 phosphoglycerate now undergo dehydration or loss of water to form phosphoenol pyruvate or 2 phosphoenol pyruvate plus water here release here. Now next is the active generation. Now look at now here the phosphoenol pyruvate now react to the ADP. Look at this is not pyruvate, this is phosphoenol pyruvate. The phosphoenol pyruvate now look at now here is our another bond is present here. This is the double bond there. Now these 2 phosphoenol pyruvate now react with ADP. ADP having 2 molecular phosphate. Then look at this phosphate from the carbon number 2 now moves to the ADP. ADP become ATP. So finally we are having the pyruvate or pyruvic acid become formed here. Look at now the 2 molecular ATP formed in the step 10 and 2 molecular ATP now formed in the step 7. So finally we are having the 4 molecular ATP form. In the step 6, we are having the 2 molecule of NAD S plus form. This is the calculation of the glycolysis. Okay. Hello.
Yes, sir.
Yes, sir.
And then now come to our the flow chart. Look at the out of the 10 steps of the glycolysis, the step 1, step 3, and step 10 are irreversible. Always you have to remember. Always step 1, step 3, and step 10 are irreversible. So there is only single arrows there. But besides all steps are reversible. Okay. Hello.
Sir.
Yes, sir.
Okay. So now here, look at now we have discussed about our the what? Now Krebs cycle or the bicarbonyl acid cycle. Look at, and before the pyruvic acid entered the tricarboxylic acid cycle or Krebs cycle, this pyruvic acid need to be converted into acetyl coenzyme A. So this pyruvate during conversion into acetyl coenzyme A, it undergo simultaneous oxidation and decarboxylation. Means the it release carbon dioxide as well as it release the 2 electron and a 2 proton. After this pyruvate release, the 2 electron and 2 proton and 2 molecule of carbon dioxide, and 1 molecule of carbon dioxide. Now, and whatever the group left is known as acetyl group. Look at, this is the pyruvate. Now this pyruvate lose. Look at, this is the terminal CO2. So for this CO now become released in the form of CO2. Now it has lost the again 2 electron and a 2 proton to form NAD S plus. And what left here? The 2 carbon compound left here right now. Now this is not acetyl group. Acetyl group now react with the coenzyme A. Coenzyme is a complex molecule. To form the acetyl coenzyme A. Acetyl coenzyme A is a 2 carbon compound. Then don't consider C as 1 carbon. This is not a carbon. This is a name, coenzyme. This is a complex biomolecule, complex organic molecule. Okay. To form the acetyl coenzyme A with the release of NADS. We had 2 molecule of pyruvate. So finally, the 2 molecule of acetyl coenzyme A and 2 molecule of NADS+ have been formed. Okay. Hello.
Yes, sir.
Yes, sir.
Okay. Now, next come to the Krebs cycle. So the Krebs cycle which occur inside the mitochondrial matrix. In case of the Krebs cycle, the acetyl coenzyme they are completely oxidized into our carbon dioxide and a FADH2 and a NAD and a NADS+. Or whatever the electron which become released during the Krebs cycle that become accepted by FADH2 and a NAD by FAD and a NAD+. So FAD is converted into FADH2 and the NAD+ converted into NADS+. So come to the how the acetyl coenzyme is oxidized in the Krebs cycle. So first we have to discuss about the steps of the Krebs cycle. So the first step is the formation of citric acid, and already inside the mitochondrial matrix already we had OAA, that is the oxaloacetic acid. So the acetyl coenzyme now react with the oxaloacetic acid in presence of enzyme citrate synthase to form citric acid, and a coenzyme remain free. So to form the citric acid. So the citric acid contain 3 carboxylic group. So it is known as tricarboxylic acid cycle, being having a 3 carboxylic group. Okay. Hello.
Yes, sir. Yes, sir.
Then next step is the dehydration. Now the citrate or citric acid undergo dehydration to form cis-aconitic acid. Next is the rehydration. This cis-aconated or aconitic acid again react with water, undergo rehydration to form isocitrate. So finally, by the dehydration and rehydration, the citric acid become isocitrate or isocitric acid. Next is the dehydrogenation or oxidation. Now this isocitrate undergo dehydrogenation or oxidation to form alpha ketoglutarate. Look at, and here actually here this step is the dehydrogenation and decarboxylation. So that then due to the dehydrogenation and decarboxylation, and then one carbon dioxide become released here, and one NAD plus is reduced into NADH plus S plus, and a alpha ketoglutarate is formed. This alpha ketoglutarate contains 5 carbon atom because one carbon atom of the isocitrate has been lost in the form of the carbon dioxide. The next step is the oxidative. Look at now we are having this alpha ketoglutarate. Now this alpha ketoglutarate, এটা কোন উল্টাটা লিখে দিয়েছি জাস্ট গোটে মিনিট। Look at just the step here. Now this is isocitrate. Now this isocitrate first undergo what? Dehydrogenation and decarboxylation to form the alpha ketoglutarate. Now this alpha ketoglutarate, now here it undergo again dehydrogenation and react with our the coenzyme to form succinyl coenzyme with the release of the carbon dioxide. Look at now here the succinyl coenzyme that become formed. Now the succinyl coenzyme react with the water molecule to form succinate and coenzyme A, and then oxidation. Then the look at the succinate become formed and which undergo oxidation, and then look at, and here the electron acceptor is FAD to form FADH2. So and then after the oxidation of succinate to form the fumarate. Now here this fumaric acid now undergo hydration and input enzyme fumarase to form malate. Now this malate undergo our the dehydrogenation to form the oxaloacetate. So in this way we are having our again oxaloacetate become formed, and in the meanwhile the 2 carbon of the our acetyl coenzyme become released in the form of CO2. Look at one add here and one add here. Hello?
Yes, sir.
Yes, sir.
Look at here, this is our the pyruvate. Now this particular the now pyruvate first convert to acetyl coenzyme, then first step citrate is formed, then cis-aconitate, then isocitrate, then isocitrate dehydrogenate. The first step is the dehydrogenation to form alpha ketoglutarate. This alpha ketoglutarate then undergo decarboxylation to form oxalosuccinic acid, this oxalosuccinic acid, and then to form succinyl coenzyme A, and succinyl coenzyme and then succinate, then are the fumarate, then malate, then oxaloacetate, then so on. Okay, then this all about the Krebs cycle, and by the end of the Krebs cycle we are having 6 molecule of NADH for 1 glucose molecule. That there are 2 Krebs cycle. Okay, look at, and then next step is what? Next step is what? Hello? Next step is what? Next step is our the electron transport system. Getting my point?
Yes, sir.
Look at, and then at the end of our the third step, here we are having 6 molecular ATP formed, another 32 molecular ATP need to be formed, and where these 32 molecular ATP present? Where these 32 molecular ATP present? Hello? এই স্টেপ পরে পর্যন্ত আমরা এই যে 32 molecular ATP हम এবে না কোডে আছেন ওইগুলি ব্রে
NADH H plus ଆଉ FADH2 ରେ
ଏବେ କଣ ହବ କି ସେମାନଙ୍କ ଭିତରେ କଣ ଅଛି? So NADH H+ and FADH2 ସେମାନଙ୍କ ଭିତରେ ସବୁ ଇଲେକ୍ଟ୍ରନ ଅଛି. When this electron are transport through the electron transport system which present on the in the our inner membrane of the mitochondria, thereby they can form the ATP. So ଏବେ ଦେଖିବା our now we have discussed about ETS. Then we have studied ETS under the following headings. Look at the essence of ETS means whatever the electron which present inside the NADH H+ and FADH2 that become oxidized to form the, or that become flow through the our the ATS to form the ATP. Okay, and then now come organization of BTS. And if we speak about electron transport system, means so but electron jai that was the electron transport system. The electron transport system of the mitochondrial membrane is consist of number of electron carriers. These number of electron carrier are organized into 4 supramolecular complex. Complex 1, complex 2, complex 3 and complex 4. हम जदि को इलेक्ट्रॉन ट्रांसफर सिस्टम ता तो तदे पूरा खन इलेक्ट्रॉन डोनर थिबा और एक्सेप्टर थिबे ता तो से माने कोन कोन माइटोकॉन्ड्रिया रे से ग्रुपिज में ग्रुप के चारटा कॉम्प्लेक्स एक छथि कॉम्प्लेक्स 1 कॉम्प्लेक्स 2 कॉम्प्लेक्स 3 कॉम्प्लेक्स 4 सब बुझियला हेलो
yes sir
देखो कॉम्प्लेक्स 1 रु इलेक्ट्रॉन टा कॉम्प्लेक्स 2 को जाए नहीं कि कॉम्प्लेक्स से 2 रु कॉम्प्लेक्स 1 जाए नहीं कॉम्प्लेक्स 1 रु कॉम्प्लेक्स 3 की कॉम्प्लेक्स 2 रु कॉम्प्लेक्स 3 and then how the electron from complex 1 will go to the complex 3 to carry electron there is a EB quinone or the quinone Q present. Got this point?
yes sir
And then from complex 3 to complex 4 the electron cannot move kind of complex 3 पाके यदि electron अछि complex 4 को जीवो complex 4 टा complex 3 पाको आस परिबो नै कि complex 3 टा complex 4 को आस परिबो नै तापर कोन complex 3 को जीवो 4 को जीवा पे हेले हमर ओटे mobile electron carrier required that is cytochrome C. So our electron transport system is comprising of four complex and a few mobile electron carrier then that has quinine and cytochrome C. Okay. Now come complex 1. Complex 1 consists of enzyme NADH dehydrogenase and NADH ubiquinone reductase who is contain phavoprotein FMN and is associated with non-hemi iron sulfur protein. Complex 2 structure. It consists of cytochrome C, oxidoreductase. Look at this structure here. Now, look at these are the then complex 4 disease, this complex 3, complex 4, and then look at and here just I'm skipping and here just I'm skipping here. Look at now come to the mobile electron carrier. There's a ubiquinone. This ubiquinone present between which complex?
complex
Ubiquinone
1 and complex 3
complex 1 2 and 3 1 2 3 3 look at this is the complete organization of BTS complex 1 complex 2 complex 3 complex 4 sometimes complex 5 का को oxygen that is the F1 F2 particle known as oxygen. Look at now come to process of electron transport. Look at this is the matrix. Look at now this NADH जो जाएगा से matrix टा matrix भीतर हमर माइटोकॉन्ड्रिया भीतर कोन अछि हमर NADH अछि जे ए NADH भीतर हमर इलेक्ट्रॉन अछि this electron now the complex 1 contain enzyme NADH dehydrogenase जे कोन करिवो NADH को भांगी की NAD प्लस करिवो दिटा इलेक्ट्रॉन बाहर करिवो दिटा प्रोटॉन बाहर करिवो now this 2 electron and 2 proton that is become come to the complex 1. Now from the complex 1 the 2 electron become transferred to the ubiquinone or the quinone then to the complex 3 then to the cytochrome C then to the complex 4 then to the oxygen. So the oxygen we are taking नाकबटे जो oxygen नौजे से oxygen आसी पहुंची कोडी this oxygen enter the matrix माइटोकॉन्ड्रिया भीतर बसे जले ए ऑक्सीजन संगे कोन हुए ए जो इलेक्ट्रॉन टा फ्लो ही गला से जले रिएक्शन करियो ओ कोन हमरो O2 माइनस हे जीवो दिस O2 माइनस पीक H plus to form the water. तो बुझियला हेलो hello
yes
getting
sir
the point look at and similarly our FADH to have been formed this FADH to कोड हमर एई जगह रे अछि look at the FADH to इलेक्ट्रॉन नबा पे हेलो एंजाइम दरकार से अछि कोडी कॉम्प्लेक्स 2 रे अछि FADH to भांगी दबो ताले FAD बाहर पड़ला आ H2 वाला H2 माने 2 इलेक्ट्रॉन 2 प्रोटॉन दीड जाका प्रोटॉन को छाड़ देला मैट्रिक्स की आ से इलेक्ट्रॉन नै गेला this the 2 electron from the complex 2 and then and follow your the red arrow move to the quinone then quinone to complex 3 complex 3 to cytochrome C to complex 4 complex 4 to oxygen. Now tell me when oxygen gains 2 electron what would be the charge of oxygen? when oxygen gains 2 electron, what would the charge of the oxygen? Hello. अरे ऑक्सीजन जदी 2 इलेक्ट्रॉन पाइला तर चार्ज करते हबो तमे देखो ऑक्सीजन केतेटा इलेक्ट्रॉन अछि केते प्रोटॉन अछि
2टा इलेक्ट्रॉन 2टा प्रोटॉन थिला
ऑक्सीजन रो
ऑक्सीजन रो
8टा इलेक्ट्रॉन 8टा प्रोटॉन तार चार्ज 0 अछि वर्तमान कॉम्प्लेक्स 4 कोन गला ताको देइला 2टा ऑक्सीजन तो कोन सॉरी ताको देइला 2टा इलेक्ट्रॉन 8टा प्रोटॉन नला 10टा इलेक्ट्रॉन ला चार्ज कोन हिजो O2 माइनस
O2 माइनस
तापरे कोन करियो देखो आमी देखो गोटे हाइड्रोजन एटम इज इक्वल टू 1 इलेक्ट्रॉन और 1 प्रोटॉन ओइगा ऑक्सीजन पाके कोन अछि ऑक्सीजन पाके हमर जेबे ना 2टा इलेक्ट्रॉन एक्स्ट्रा अछि से कोन करी मैट्रिक्स अछि मैट्रिक्स भीतर कोन अछि हमर मैट्रिक्स भीतर हम वाटर अछि एटी कोन किछु वाटर H+ आउ हमर OH माइनस हिथिबे से कोन करियो वाटर माने H+ टा से निजळी भिडी दबो के ऑक्सीजन टा तले कोन होबो ऑक्सीजन पाके 2टा इलेक्ट्रॉन अछि गोटे इलेक्ट्रॉन प्लस गोटे प्रोटॉन मिसिले कोन हिजो गोटे हाइड्रोजन आउट इलेक्ट्रॉन आउट H+ मिसिले आउट हाइड्रोजन हिजो 2 फ्रॉम द वाटर ओके
यस सर
so देखो then the route through which the NADH oxidized is known as the route 1 the route through which FADH2 become oxidized known as route 2 got this point नाली डाला route 2 आउ पळा डाला route 1 लगा देखो route look at route 1 complex 1 ubiquinone complex 3 cytochrome C complex 4 oxygen look at it this oxygen when gets 2 electron 2 O2 minus plus 2 proton is equal to water okay
yes sir
this is the route 2 complex 3 ubiquinone complex 3 cytochrome C complex 4 then O2 minus S plus S the 2 proton water look at now here I had told you and then when to the electron transfer system electron flow our ATP form or here the electron already has transferred our the oxygen has been reduced into water oxygen ebe water he gala तले छुआ को आमे नाक बाड जो ऑक्सीजन टा नौ सेटा कार्बन डाइऑक्साइड की बाहर ना वाटर की हमर ना बॉडी भीतर रहि जाय
कार्बन
कार्बन
डाइऑक्साइड
डाइऑक्साइड
बाहर
अरे पिला मन तो कहली बरा बे नाक बाड ऑक्सीजन की एठी माइटोकन एठी अछि टी बे ए ऑक्सीजन टी अरे नाक बाड ऑक्सीजन आसला की ए ऑक्सीजन टी तले ए ऑक्सीजन तो कन्वर्ट कोडी गेला
वाटर को
तार जो कार्बन से उड़ी गला से हला ग्लूकोज कार्बन डाइऑक्साइड ग्लूकोज कार्बन टा बुझ पेला
सर
आइडिया क्लियर तो मैक्सिमम टाइम दी स्टूडेंट्स हैविंग दिस प्रॉब्लम हमें जो ना नौ ऑक्सीजन सेटा कार्बन से की सेटा ना बाहर पड़ी जाउची इज इट राइट
sir
okay look at now here the electron transport has been completed what 8 no ATP form एपन तो कौन सी ATP तैयारी है एबे देखियो ATP केम तैयारी होची look at actually the complex 1 complex 3 complex 4 they have our proton pumping activity when electron from complex 1 moves to the complex 3 amount of energy released that cause pumping of 4 proton from the mitochondrial matrix into the perimitochondrial space एति वाले भाबी रखो जते बोले इलेक्ट्रॉन टा हमर कोइ complex 1 रु ए ubiquinone नु गला ये complex 1 कोन करची माइटोकॉन्ड्रिया भितरु 4टा प्रोटॉन नै की ए पेरीमाइटोकॉन्ड्रियल स्पेस ने छाड़ दे के आछी बुझ पेलो जते बोले ये complex 3 रु जाउची साइटोक्रोम सी की complex 3 कोन करची 4टा प्रोटॉन धरची कोडी मैट्रिक्स रु छाडुची हमर पेरीमाइटोकॉन्ड्रियल स्पेस रे जते बोले हमर complex 4 रु जते इलेक्ट्रॉन ऑक्सीजन को आछुची complex 4 having proton pumping activity it has the proton pumping activity is transport the 2 proton from the matrix to the perimitochondrial space so मतलब route 1 रे how many proton pumped route 1 रे केतरा प्रोटॉन पंप होइछ हेलो route 1 रे समुदाय केतरा प्रोटॉन पंप होला how many proton have been pumped through route 1 of electron transfer हेलो अरे complex 1 तो 4टा पंप कला कॉम्प्लेक्स 3 तो 4टा पंप कला कॉम्प्लेक्स 4 तो हमरो 2टा पंप कला समझ केते 4 4 8 10
10 टा
देखो एबे रूट 2 देखो नाली देखो लो कॉम्प्लेक्स 2 रे इलेक्ट्रॉन कोड गला ऑक्सीजन कोड हम क्यू को गला कॉम्प्लेक्स 2 लाके प्रोटॉन पंपिंग एक्टिविटी तार राही जो कॉम्प्लेक्स 3 रो जो साइटोक्रोम सी को 4टा प्रोटॉन पंप कला ए कॉम्प्लेक्स 4 जो ऑक्सीजन को आसला देन द 2 प्रोटॉन पंप कला समझे रूट 2 रे मत को रूट 2 रे केते प्रोटॉन पंप कला
6 टा
6टा ताले रूट 2 रे ऑक्सीडाइज किए हुए NADH ना FADH2 रूट 1 रे ऑक्सीडाइज किए हुए रूट 2 रे ऑक्सीडाइज किए हुए
रूट 1 रे NADH
અને रूट 2 रे FADH2 तार
FADH2
दशमन मन कर जो जो प्रोटॉन को पंप होला से प्रोटॉन जते बेले फेरिवो कोडू फ्रॉम दी पेरिमाइटोकॉन्ड्रियल स्पेस माने माइटोकॉन्ड्रिया इनर मेम्ब्रेन आउटर मेम्ब्रेन भीतर अछि के ना प्रोटॉन टा जते बेले से फेरियो भीतर को सेते बेले हम एक्टिव तयारी हो look at the proton have been formed when this proton now return back into matrix जितरे से माइटोकॉन्ड्रिया भीतर पसिबो सेते बेले हम एक्टिव तयारी हो then according to old concept कहूची when 3 proton enter into the matrix amount of free energy available that cause formation of 3 molecular ATP sorry then 1 molecular ATP old हम कोन old concept कहला जेतेबेले हमर जो जेतिको प्रोटॉन पंप हेतिले पेरिमाइटोकॉन्ड्रियल स्पेस को जेतेबेले से माने एंटर करूचंती फॉर 3 प्रोटॉन 1 molecular ATP फॉर्म तले रूट 1 ने समुदे हमर केतरा प्रोटॉन पंप हेची
10 टा
10 टा केतरा एटीपी तयारी हो
3 टा
3 टा रूट 2 रे FADH2 ପାଇଁ
6 टा हेची 2 टा हबो
2 टा हो को दिबे so 4H then okay and then to explain this formation of ATP the Peter Mitchell proposed the chemiosmotic hypothesis for that he got Nobel Prize in the year of 1978 to explain how this ATP formed so this is the Peter Mitchell this is the F1 F2 particle then don't say F1 F0 this is F1 F2 particle O for oligomycin sensitive and through this proton transport occur this is the this okay this is the expanded view ATP okay
yes sir
Yes, sir.
so that you look at and then and here and whatever we have discussed this is the data here look at and besides then I had told you and then the NADH H+ that formed in the glycolysis then it look at it cannot directly enter into our the mitochondria so that here this NADH H+ that formed in the glycolysis it has to enter into the mitochondria it has to follow some subtle system so and then the NADH H+ of the glycolysis it had to transfer the electron to the either to the glycerol phosphate subtle system or to the aspirated malate subtle system and then when NADH H+ of the glycolysis oxidized to the glycerol phosphate subtle system there 2 molecular ATP form when oxidized to the aspirated malate subtle system 3 molecular ATP form only just you have to remember एथिकल मन रखता जो माइटोकॉन्ड्रिया जो हमरो NADH H+ जो हम ग्लाइकोलाइसिस तयारी हेची से माइटोकॉन्ड्रिया भितर पस परिबे नाही से कोन करिवे ना माइटोकॉन्ड्रिया को जाय आसीबे ना माइटोकॉन्ड्रिया रो इनर मेम्ब्रेन जाय आसीबे इनर मेम्ब्रेन को से दे दे की पळे भाबी दे ओटा इनर मेम्ब्रेन रे ओटा सटल सिस्टम अछि सपोज दिस ग्लिसरॉल फॉस्फेट सटल सिस्टम आ से कोन करिवे से जो NADH जो तयारी हेतिबो साइटोप्लाज्म रे से आसीबो माइटोकॉन्ड्रिया रो जो इंटर मेम्ब्रेन स्पेस अछि से जाय आसीबो से जाय आसी की आ सपोज आरि पर बसची ओटे प्रोटीन बसची ताको जे हमें ग्लिसरॉल फॉस्फेट ताको से दे दी पड़िबो दिटा के इलेक्ट्रॉन जदि जो दिटा इलेक्ट्रॉन पाइला जदि से FADH को दबो इलेक्ट्रॉन ताले FADH नै कि जीवो हमर ETS रो 6टा प्रोटॉन पंप हबो दिटा टी बारिबो जदि जो इलेक्ट्रॉन टा जो NADH आनी के आसी जदि सी एस्पिरेट मालेट सिस्टम देला ताले सी दबो काहे को सी दबो आउडे NAD से आउडे दबो NAD+ को दबो माइटोकॉन्ड्रिया भितरे to form 3 molecular ATP सब बुझिला डा हेलो
हाँ हाँ सर
क्लियर तर मते कहले बे ना कोन पे हम माइटो कोन पे ग्लाइकोलाइसिस रो NADH टा 2 molecular ATP ଦଉଛି କାହିଁକି ସେ ମାଇଟୋକଣ୍ଡ୍ରିଆ ଭିତରୁ ପଶି ପାରୁନି ସେ କଣ କରୁଛି ସେ ଯୋଉ ଗ୍ଲାଇକୋଲିସିସ୍ ର NADH ଟା ଆସୁଛି କୋଉଠି କି ଆରେ ଆସୁଛି କୋଉଠି ଏବେ ନା ପେରିମାଇଟୋକଣ୍ଡ୍ରିଆ ସ୍ପେସ୍ ଭିତରକୁ ଆସିବ ଇନର ମେମ୍ବ୍ରେନ ରେ ଗୋଟେ ସଟଲ ସିଷ୍ଟମ ଅଛି ତାକୁ କହନ୍ତି ଗ୍ଲିସରଲ ଫସ୍ଫେଟ ସଟଲ ସିଷ୍ଟମ ତାକୁ କଣ କରିବ ସେ ଇଲେକ୍ଟ୍ରନ ଦେଇ ପଳେଇବ ସେ ଗ୍ଲିସରଲ ଫସ୍ଫେଟ ସଟଲ ସିଷ୍ଟମ କାମ ସେ ମାଇଟୋକଣ୍ଡ୍ରିଆ ଭିତରେ ଯୋଉ ଏଫଏଡି ଥିବ ତାକୁ ଦେଇଦେବ ଇଲେକ୍ଟ୍ରନ ଏବେ ଏଫଏଡି ପଳେଇ ଯିବ କଣ ରୁଟ୍ 2 ପଳେଇବ ରୁଟ୍ 2 ପଳେଇ 6ଟା ପ୍ରୋଟନ ବମ୍ ହବ 2ଟା ATP ତିଆରି ହବ Got this point?
sir.
ये थिला तमरो दिस द ETS इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम and then besides and we are having
सर आपण हमको नोट टिके देले
मु तो दे दी थिले से छुआ जति पठै थिले अरे पठै थिले का सब माने पठै छिना नोट तुम सब जे देथिला
हां सर
देखो छुआडा आ गोटा हमर छुआ अछि गोटा फर्मेंटेशन अछि भाबिबो जते बेला ऑक्सीजन हम एनवायरनमेंट नाही व्हेन लुक एट फर्स्ट दे मन थिबो दी ना ग्लाइकोलिसिस इज कॉमन टू बोथ एरोबिक एंड एनारोबिक रेस्पिरेशन ग्लाइकोलिसिस कोन अछि हमर एरोबिक रे हो एनारोबिक हो ग्लाइकोलिसिस हैला परे कोन हो हमर पायरुबिक एसिड तयार हे जिवो जदी हमर एनवायरनमेंट ऑक्सीजन अछि ताला हमर क्रेब साइकिल हो ETS हो जदी ऑक्सीजन नाही ताला फर्मेंटेशन हो फर्मेंटेशन दी प्रकार गोटे अल्कोहलिक फर्मेंटेशन गोटे लैक्टिक एसिड फर्मेंटेशन अल्कोहलिक एसिड फर्मेंटेशन कोन हेबे तो पायरुवेट फर्स्ट जीव कोन बे डीकार्बोक्सिलेशन जीव टू फॉर्म एसिटाल्डिहाइड this acetaldehyde become reduced by NADHS plus that was released in the glycolysis to form ethyl alcohol and NADH so ethyl alcohol is formed that found in yeast ଆଉ कोन होची हमर lactic acid fermentation होची then you might have seen during the cricket match there is some muscle cramp occur in the players देखते पिला मन सब क्रिकेट मैच होची रोहित शर्मा गोडटा मोडी गेला चाली पइले 100 बाडी सेला पर देखियो एटा मसलटा हार्ड हे जे हेलो अरे ऐसो नो
Yes, sir. Yes.
तले कोन पे जते बेले हम अधिक परिमाण जते बेले दौड़ कोन हुए हम बॉडी रो ऑक्सीजन टा शॉर्टेज हुए तले बॉडी रो ऑक्सीजन शॉर्टेज कोडा मन मसल ऑक्सीजन पाएनी सेही क्षेत्र कोन हुए हमर ग्लाइकोलिसिस कंटीन्यूअस लव ऑक्सीजन न हो बचे एने रोहित शर्मा को मैं छक्का मारिबी आ बॉल कम अछि बट धोई से हे गेलानी ता बॉडी के ता नाको आ ऑक्सीजन सप्लाई करि पामने स्टिल देन से चाहू मु छक्का मारिबी तारे कोन एनर्जी दरकार से जगह हमर कोन होबने आ हमर एरोबिक रेस्पिरेशन होबने क्रेब साइकिल होबने ETS किछि होबने ताले खाली हम ग्लाइकोलिसिस होबो for 1 molecule of glucose 38 molecular ATP formed due to the our aerobic respiration when oxygen present but oxygen deficiency होची oxygen नहीं पारुनी but ताहु जो छक्का मारी ताले कोन हमर एबे ना ग्लाइकोलिसिस खाली होबो when 1 molecule of glucose undergo like under the glycolysis only 2 molecule of ATP form माने net gain so it has to manage the breakdown of a large amount of the glucose when the large amount of glucose become break in the muscle that cause the formation of huge amount of pyruvate this huge amount of pyruvate now converted into lactate now it undergo reduction to form the lactate and then or form the lactic acid when the lactic acid deposited out the muscle that becomes stiff that become hard so it is very difficult to work okay hello
yes sir
next is our pentose phosphate pathway and just you have to remember that generally our body contain hexo sugar but on DNA RNA contain ribose sugar or pentose sugar the process by which these pentose sugar are formed from hexo sugar and this is our the pentose phosphate pathway look at this is our the hexo sugar the glucose 6 phosphate is present here continuously our the pentose sugar which are also formed here just you have to remember these things okay and you can also directly study it from the node also here there are some steps are present here just come to our significance of pentose phosphate pathway it provides alternative route of carbohydrate breakdown it generate then due to the pentose phosphate pathway carbohydrate and then in our body the carbohydrate breakdown occur NADPH also form it provides ribose sugar for synthesis of nucleic acid it play important role in fixation of carbon dioxide in photosynthesis because the Rubisco, the ribulose 1,5-B phosphate, that was the carbon dioxide acceptor. Just a minute. So that the ribulose 1,5-B phosphate, that is the carbon dioxide acceptor of the Kelvin cycle, that is also formed by this pathway. Just you have to remember, you have to practice this pathway. It is a long question for you. Hello?
Sir.
Hello, getting my point?
Yes, sir.
sir.
Look at here about the next part of the enzyme, and just to discuss. Enzymes are biochemical catalyst. They accelerates biochemical reactions without changing the equilibrium. That the enzyme माने हम बॉडी भीतर कोन देखिबो ना जे कोन जोड रिएक्शन हमर बॉडी भीतर होछि को स्पीड रहउछि ता करूछि कि जे करूछि सेला एंजाइम बुझ पाइलो
Sir.
and then look at first come to this structure of enzyme, and when anyone speak to enzyme, these structures should be common in your mind. For example you take one reaction. Suppose inside your body the glucose is converted into fructose. Let's example. So glucose cannot directly convert into fructose. So that first glucose come and bind with the enzyme. Let's examine enzyme the X. Suppose the glucose is bind here. Let's example. Suppose this glucose is bind here of the enzyme. After that, the enzyme break the bond of the glucose and convert the glucose into what? And convert glucose into fructose. So, in absence of this enzyme, the glucose can convert it into fructose. This is the fructose. But the rate is very slow. But in presence of enzyme, so that a huge amount of glucose can be converted easily into fructose. So, the function of enzyme to accelerate the reaction. In absence of enzyme, the reaction can occur, but the rate is very slow. But in presence of enzyme, the reaction occurs very fast rate. What is point? What enzyme which cannot change the equilibrium? Oh, always you have to remember. So first come to the structure of enzyme. Generally enzymes are chemical composed of protein. And when an enzyme is completely composed of protein, is known as the simple enzyme. Hello, getting my points?
Yes, sir.
Yes, sir.
And when an enzyme is completely consisted of protein, is known as the simple enzyme. But some enzyme which require a protein part as well as a non-protein part for function, then such enzymes are known as conjugated enzymes. The protein part of enzyme known as apoenzyme, and the non-protein part is known as cofactor or posterior group. When the cofactor, look at, and when the cofactor is a complex organic molecule that is loosely attached to the enzyme, is known as coenzyme. When the cofactor is tightly attached with the enzyme that is not separable, is known as posterior group. What is point? Enzyme है ताले एंजाइम पूरा कंप्लीट प्रोटीन नहीं गिरो पूरा देखो पूरा नाली पाटा रहिबो पूरा ई हला हमर सिंपल एंजाइम एते बोले गोटे एंजाइम रे प्रोटीन पार्ट थियो नॉन प्रोटीन पार्ट थियो ताको हम कहबा कंजुगेटेड एंजाइम प्रोटीन पार्ट का अपो एंजाइम कहबा ते नॉन प्रोटीन पार्ट का कहबा कोफैक्टर में भी ए कोएंजाइम में भी पोस्टेड ग्रुप कोएंजाइम केते बोले कहबा जेते बोले ई जो कोफैक्टर ता जे नॉन प्रोटीन पार्ट ता पूरा कॉम्प्लेक्स ऑर्गेनिक मॉलिक्यूल हबो से लूजली अटैच हे रो पछे छाडी जे करथियो बट द पोस्टेड ग्रुप when the non-protein part is tightly bound to the enzyme, is known as a posterior group. What is point? Look at the site of enzyme where the substrate bind. In case of enzymatic reaction, the reactant known as substrate. Suppose here glucose is converted to fructose, glucose is the substrate, fructose is the what? Product. What is point? Hello.
Sir.
Yes, sir.
Look at this is the now binding site, this is the active site of enzyme. Okay, and then sometimes the enzyme may be monomeric. Monomeric means consist of only one subunit. Sometimes it may be polymeric. This is the structure of RNA polymerase. RNA polymerase consists of about the 5 types of subunit: omega, beta, beta prime, sigma, and alpha. But not the number is, look at this is the here being is composed of the number of subunits is known as what? About the oligomeric enzymes. Look at here what the types is 5, but number is 6. 1, 2, 3, 4, 5, 6. Look at the next is the mechanism of enzyme action. To explain mechanism of enzyme action, we have to study it under the 2 heading: formation of enzyme substrate complex, and then the lowering activation of energy. At the mechanism of enzyme action implies how an enzyme works. Enzyme केमिति काम करे. We have to study under 2 heading: formation of enzyme substrate complex and then lowering activation energy. then गोटे एंजाइम काम करबे पे हेले प्रथम से सबस्ट्रेट संग बाइंड करबे पड़िबो, गोटे रिएक्शन संग गोटे चिना रिएक्टेंट संग से बाइंड करबे पड़िबो. To explain formation of enzyme substrate complex, the 2 theory have been proposed. One is lock key model, second is the induced speed model. In the lock and key model, you consider enzyme as lock and substrate as key. As a particular key is required to open a particular lock, when a particular substrate is bind to particular enzyme, that causes formation of enzyme substrate complex. So this substrate in the further step converted into product. For this point,
Yes, sir.
the next is the induced speed model. According to the induced speed model, the enzyme do not have the pre-formed active site. Enzyme रो active site नहीं कि जो जगह रे हमर bind हे पर substrate ता जेतबे substrate जाई bind होबो सेतेबेले active site रे design हो already active site अछि, but that is not precept. When substrate bind, that cause conformational complementary change at active site. So then the enzyme substrate complex is formed. भाभी जे गोटे गाड़िया अछि ओना प्राय पट्टू माटी भाभी जे गोटे गाड़िया सुखी जाइछि कादु ओरा के तुम देखा जाउछि गोटे पर्टिकुलर जगह कोना हला भाभी दियो से भाभी दियो तान गाड़ीटा पूरा हला एंजाइम गोटे पर्टिकुलर जगह कोना भाबो सेटा एक्टिव साइट सेही पर के प्लेन हे गछि तो टेक्का दो टेक्काटा सबस्ट्रेट टेक्काटा पकइलो सेही जगह टेक्काटा ने पइला ता भितर पसि जीवो तार तो से गोटे एक्टिव साइट ओलिता शेपटा डिजाइन ही नथिला सबस्ट्रेट बाइंड कर टेक्काटा पइला पर शेपटा डिजाइन हे गेला ताले सेटा तुमरा इंड्यूस स्पीड मॉडल. तो बुझिले रे?
Yes, sir.
And then ये थला हमरो so that we can study mechanism of enzyme action under 2 heading. One is कोना our formation of enzyme substrate complex, and the second one is and lowering activation energy. So first we have discussed about what is activation energy, and then look at, and then to commence a reaction, to start a reaction, there is some amount of energy required. The amount of energy which is required to start a reaction known as activation energy. What is point? So now we have to explain activation energy by taking an example. For example, you are having 2 firewall. तुम घरे दिटा चुली अछि. गोटे चुली कोन करला गोटे ओदा काठ भर्ती करला छाड़बे ये कोन करले गोटे चुली रे तमे गोटे सुना सुखिला काठ भर्ती करला. So as soon as you entered the dry wood into the fire hole, does it suddenly burn or it takes some time to burn?
It takes some time to
Takes
burn.
some time to burn.
One time, it first takes some heat energy to first hot. Kichi energy nala garam hala. When amount of heat energy absorbed, it is equal to burning temperature. After that, it start to burn. Okay. So this amount of energy, so they are burning with the reaction. Burning पूर्व जेतिक energy नला जलिया पाई, that is the activation energy. So if we place a wet wood into the fire hole, more amount of energy it will require to burn as compared to our the dry wood. है नहीं? तो चला हमर ओदा काटा परिया से अधिक एनर्जी नबो काई प्रथमे से एनर्जी नै पानी उड़ेबो पानी उड़े से परे सुखिला हबो सुखिला पर एनर्जी नै चलिबो. So here activation energy is more. Yes or no?
Yes, sir.
Sir.
For this point?
Yes, sir.
Yes, sir.
Now tell me, when activation energy is more, the reaction is faster or slow? When activation energy is more, reaction is fast or slow?
Slow.
When activation energy is more, reaction should be slow. काठटा हमर ना लेट जल्ला उदा काठ activation energy जते कम होबो reaction समय सेते speed
Faster habo.
the enzyme कोन करछि enzyme speed the reaction by lowering the activation energy देखो in absence of enzyme the activation energy this much look at this is the na red or in presence of enzyme activation energy is very less. Okay.
Yes,
Yes,
sir.
sir.
Next is the Michaelis-Menten equation. This is the equation that was given by Michaelis and Menten to explain at which speed our the enzyme catalyzing reaction. Our enzyme टा केते speed रे reaction टा catalyze करूची आउ तार जो से speed टा हमर enzyme रे concentration और substrate concentration में केम depend करूची. For that they proposed an equation. And finally, we have now this is the derivation. It is not possible to discuss here in details. And the final equation we have got this is the V0. V0 is the initial velocity. V max is the final velocity. Is the maximum velocity. S is the substrate concentration. Km is the Michaelis-Menten constant. This is the equation by which we can also calculate. And then the enzyme inhibition. Man enzyme रा काम करिबा बंद करिबा को enzyme inhibition. The process by which enzyme activity slow down or stop is known as enzyme inhibition. The agent which cause enzyme inhibition called enzyme inhibitors. Enzyme inhibition may be reversible, may be irreversible. Reversible माने कोन हो थरे हमर enzyme अछि inhibitor से bind हे गेला परे enzyme रा temporarily inactivity होबो. If हमर when we remove the inhibitor then enzyme become free again and it again can catalyze the reaction known as reversible. In irreversible what happen after once the inhibitor bind then the enzyme remain permanently deactivated or inactivated known as irreversible. The reversible may be competitive. In competitive inhibitor shape टा पूरा substrate संग समान हबो जहा फळे रे कोन हेबे inhibitor और substrate गोट enzyme रे active site पे से माने compete करिवे. And then uncompetitive. In uncompetitive inhibition, an inhibitor binds at a site distinct from substrate. Substrate से अलग जगह से कि ना bind करिवो. Look at these are the next is our the non-competitive inhibitions. The pure it may be the following type pure non-competitive and it may be the mixed non-competitive inhibition. The figure is there and you have to and then I will send this particular tip and note to you. You have to go through this note. If there is any problem, then you may ask over my WhatsApp. Okay. Now come this is the irreversible. Hello.
Yes, sir.
Yes, sir.
Look at in case of the irreversible, what happened? This is the enzyme. Here, the active site. This is the inhibitor. This inhibitor permanently bind to the enzyme by covalent bond. So enzyme is completely deactivated here. This inhibitor, which look at this inhibitor bind with the active site of enzyme because the active site where serine is present. Serine is amino acid. This inhibitor which directly bind to this particular the serine. So this serine is very much essential for activity of enzyme. When the serine is blocked, this enzyme is completely blocked. This is a group specific inhibitor. This is a comes under irreversible. Means after binding the inhibitor, enzyme completely deactivated. And the next the substrate analogous. Suppose one example when the shape of the substrate is similar to the shape of the inhibitor. And in this case what happened? And this inhibitor come and bind to the active site of enzyme permanently. That block the complete active site and that bind by forming the covalent bond. In this case, the enzyme become inactive. Suppose this enzyme are also called active site contain the glutamate. But one compound known as 3-bromo lactose. Sorry, what is that? 3-bromo acetyl phosphate. Now first bind at here to this now glutamate. This glutamate now become completely linked here. This enzyme is inactive because this glutamate is essential for the function. Next is the suicide inhibitor. This is also a reverse type of inhibition. Suicide inhibitor means what? Look at and here the shape of the inhibitor is same to shape of the substrate. And enzyme is unable to recognize which is substrate, which is inhibitor. Now this inhibitor bind to the enzyme, and then this enzyme start to convert substrate to the product. But meanwhile, this then some intermediate of this particular conversion remain act as inhibitor, complete deactivate enzyme. So बुझी पाए हेलो ना कौन कहले एटा देखो आम जो कहबा कि सुइसाइड इनहिबिटर तार तो गोटे एंजाइम अछि फॉर एग्जांपल दिस इज एन एंजाइम एंजाइम सबस्ट्रेट आसला से एंजाइम रे जानि परला कि मोर पे कोन सपोज दिस द इनहिबिटर देखै पे जेमतिया सबस्ट्रेट देखै पे सेमतिया तो जानि परला ने कि एटा इनहिबिटर ना सबस्ट्रेट ता से से ना बाइंड कर देला बाइंड करसला परे से सबस्ट्रेट एका बेले कन्वर्ट हे परिबो ने कि हमरो ता प्रोडक्ट टू सपोज द सबस्ट्रेट इज अंडरगो सम मॉडिफिकेशन फर्स्ट स्टेज गला सबस्ट्रेट कन्वर्ट इनटू S1 हैला सबस्ट्रेट पर S2 हैला सबस्ट्रेट कन्वर्ट S3 हैला S3 पर जाय से कोन हबो ना प्रोडक्ट हबो सपोज व्हेन दिस इनहिबिटर कन्वर्टेड इनटू S1 एंजाइम काम करछि S2 हैला बेले तो S2 एक्ट एज इनहिबिटर आफ्टर दैट इट कंप्लीट डीएक्टिवेट एंजाइम बट जेते से इनहिबिटर बाइंड हेतला से एंजाइम की डीएक्टिवेट करि पानतला बट एंजाइम जेते से इनहिबिटर टा को भाबी कि निज सबस्ट्रेट भाबी कि प्रोडक्ट कन्वर्ट करे चेष्टा कला तार तीनटा फॉर्म हैला S1 S2 S3 हैला पर जे प्रोडक्ट हबो से इनहिबिटर जो सेकंड फॉर्म टा जो चेंज हैला एंजाइम द्वारा से उडा का एक्ट एज इनहिबिटर एंड इज कंप्लीट डीएक्टिवेट एंजाइम हेलो बुझ पेले
Yes, sir.
अरे बुझु ना खाली एस रे कहजो ठीक बुझियो तो
हाँ सर बुझी अछि
हम जो फीडबैक इनहिबिशन सपोज कहबे कि गोटे रिएक्शन अछि गोटे रिएक्शन पे हम पांचटा एंजाइम दरकार हैला सपोज ए बी सी डी ई हैला सपोज हैला देखो ए इटे कोन देखो ए हैला हमर ओडला गोटे सबस्ट्रेट अछि ए ऊपर सबस्ट्रेट ऊपर एंजाइम 1 ऐड कला फिर ता प्रोडक्ट बी हैला बी जो हैला बी हैला का पे ई टू पे सबस्ट्रेट सेकंड एंजाइम ता बी ऊपर ऐड कला ता कन्वर्ट कला सी की सी ऊपर ऐड कला एंजाइम 3 ता कन्वर्ट कला डी की डी ऊपर ऐड कला एंजाइम 4 ता कन्वर्ट कला ई की हमर को सेल कोन दरकार ई दरकार सपोज भाबिबा बे कंसंट्रेशन ऑफ हमर ई टा सेल भीतर पे बहुत बढ़ी गेला बट द सेल नाउ नो लॉन्गर रिक्वायर्ड आवर ई सेल ई दरकार नै सेति बेले कोन हबो ए ई टा हमर एंड प्रोडक्ट टा ई आसीबो फास्ट एंजाइम से बाइंड हो जो कोड बाइंड होबो ए एक्टिव साइड बाइंड होबो नै ए जगह बाइंड होबो नै बाइंड न होय कि इट बाइंड टू अनदर साइड ता पछ बॉडी की जेकोन जगह आउटसाइड बाइंड हो जो के ई टा इज द प्रोडक्ट से कोन करे आउ एंजाइम 1 टा को काम करे पे दबो नै ओके ता को जे फीडबैक इनहिबिशन सो दिस एंजाइम 1 कैन नॉट वर्क हियर हेलो
हाँ सर
Yes, sir.
got this point
यस सर
that's the nomenclature of enzyme look at now this is the classification of enzyme and then the enzyme commission recognized the 6 classes of enzyme oxidoreductase those enzyme which cause oxidation and reduction reaction transfer is AB plus C A and BC when the enzyme which cause group transfer known as transferase hydrolase the enzyme which break the bond by using water lysase the enzyme that introduce double bond by group transfer then isomerase the enzyme which undergo isomerization ABBA ligases the enzyme which join known as the ligase A plus B plus ATP to form AB here A is 1 B is 1 but here AB is complete 1 okay active site you knows the site of enzyme where substrate bind known as active site coenzyme are the complex organic molecule they are loosely bind to the enzyme okay the coenzyme examples are NAD plus NADP plus FMN FAD okay hello
Yes, sir.
then all are the this is all about our the respiration as well this I think tomorrow will be the last class.
କାଲି ନେକ୍ସଟ ପରଦିନ କ୍ଲାସ୍ ଅଛି।
Hmm?
କାଲି କ୍ଲାସ୍ ନାହିଁ, ପରଦିନ କ୍ଲାସ୍ ଅଛି।
ପରଦିନ କ୍ଲାସ୍ ଅଛି?
Yes, sir.
Just a minute. Mm-hmm. 23. Okay, I think the day after tomorrow. Okay. What is the last class? Now tell me. And as to that, and have you got something from the today's class? Hello.
Yes, sir.
Yes, sir.
Have you understood something about the respiration? Yes or no?
Yes, sir.
Yes.
Yes, sir.
Wait a minute. Okay, thank you very much. In the coming class, then we have to discuss about the phytohormone and rest of and then whatever left. Okay.
Yes, sir.
Thank
Thank
you,
you,
sir.
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 Thank you,
Sir.
Thank you, sir. Hello, Kavit. Hello. Hello.
Hello.
କିଏ ସଚିନ?
Yes.
ଫିଜିକ୍ସ ର ଯୋଉ 135 ର ନୋଟ ପାଇଛ କି?
ନାଇଁ ମୋର ଫିଜିକ୍ସ ନୁହଁ, ମୋର ସିବିଜେଡ୍।
ଓହୋ। କବିତା ପ୍ରଧାନ କିଏ?
Hello?
ହ୍ୟାଲୋ। କବିତା ପ୍ରଧାନ।
Yeah, tell me.
ଫିଜିକ୍ସ ର 135 ର ନୋଟ ପାଇଛ କି?
नहीं तो
ତୁମେ ବରଗଡ଼ର କି?
नहीं नहीं संबलपुर स्टडी सेंटर झारसुगुड़ारे रहुछु।
ଓହୋ। ସମ୍ବଲପୁରରେ କଣ NSCB କଲେଜ?
हाँ
ତୁମ ନମ୍ବର ଟା ଦେଇପାରିବ କି?
हाँ नियो
ହଉ କୁହ।
9742
9742
64
64
3550।
3550। ହଉ ଥ୍ୟାଙ୍କ ୟୁ। ଏଇଟା ମୁଁ ବି NSCB କଲେଜରେ ଇଗ୍ନୋରେ ଆଡମିସନ କରିଛି ତ ସେଥିପାଇଁ ନମ୍ବରଟା ମାରିଥିଲି।
हाँ हाँ ठीक अच्छे मते व्हाट्सअप करबो तुम्हें।
ହଉ ଠିକ ଅଛି।